Research for cloning has been around for more than 100 years. It all started with a man named Hans Adolf Edward Drake. He was a German man, and he had a doctorate, and his main his main interest was phylogeny, which is like a special branch of evolutionary theory. His first experiment, or like reported experiment, was with sea urchins, and what he did was he separated the cells by shaking the two-celled sea urchin embryos, and once they were separated, they were able to grow into a full organism. So that happened in 1885, and since then there has been plenty of more experiments. They have experimented with salamanders, frogs, rabbits, cows, monkeys, endangered animals, and a famous sheep, Dolly, that happened in 1996. And there is a lot of controversy with cloning because it violates ethic codes, so that is considered a problem at times.
So hi, my name is Stephanie Gomez, and I will be talking about the histories and the history and discoveries and breakthroughs of cloning throughout the years. And so when I was researching through it, I found a timeline out of the Genetic Science Learning Center. And with this, I saw all the different types of cloning, starting from just getting an artificial embryo to start the twinning, which is the twinning being a form of cloning, and then it goes all the way down to, I believe, 2013, when they talk about wanting to do human cloning. It didn't go on telling me about as far as how well that experiment went on, but it does tell me a lot, and it did tell me the first ever cloning, which was the embryo twinning. And so two scientists, they went to shore, they grabbed a sea urchin, shaked the two cells that were in the sea urchin's embryos, and by them doing that, the cells separated. So as soon as the cells were separated, they grew into a completely different sea urchin. But that was their form of twinning. And as this was the first ever twinning, and this was actually in 1885. So they grew into a full organism. So like I said, this was the first ever form of cloning that we did see, which was in 1885. So as you see, cloning has been around for a while. It's just little bitty things of how far scientists get into developing new ways of cloning. So I did go on and I read that in 1996 they did a cloning form, which was nuclear transfer. So nuclear transfer is a form of cloning where they take away the DNA. They take parts of the DNA to create the same, because DNA has all our information, all of our genetics. So they got that DNA. So now moving on to 1996. Again, a little bit further down in the timeline, when I first started off, from 1855 to when they were doing the embryo twinning to now when they did the nuclear transfer. And this was the first mammal created by the somatic cell nuclear transfer. So this was a very landmarked experiment as to where two scientists got a lamb, and they created transferring the nucleus from an adult sheep into eggs. And while this was happening, they kept doing this, and I believe it was over like roughly around 277 attempts that they got, where they finally produced one embryo that was carried full term and a pregnant sheep. So this baby sheep came out, and they named her Dolly. So Dolly was a very famous sheep because, as you know, like this was the first ever mammal created by cloning. Um, it really put a marking for the scientists as well. It showed different ways of cloning and how much it had developed over time, and how different uses of different ways showed how they can create the same genetic information. This still wasn't at the midpoint as to where they kept going on and on and on and on. They kept using different animals, different mammals. One that really caught my attention was the first ever cloning of monkeys. So now we're moving on to just a year later, and this would be 1997 when they did the cloning on monkeys. So 3 scientists came together and they were trying to find different ways of how to clone, and they actually found a way of doing a small electric shock. So they got a surrogate mother, of course, a monkey, and out of 29 cloned embryos, only 2 monkeys were born. And out of those 2 monkeys, one was a female and one was a male. The way and the process was similar to the way they did Dolly the sheep's cloning, but they just added the electric shock to it. It was them again trying different ways, trying to make this cloning more and more successful and letting the mammals last longer in the world. And so this again was a very important experiment because, as you know, monkeys are close relatives, and so this was showing that since monkeys are close relatives, they were able to be cloned, and it gave scientists more and more ideas as to how they can clone us. So they kept putting together different ways of cloning and trying to fix it and make it perfect. So now again, we're moving down the timeline to 2007. And this is again where they're doing experiments on monkeys. So researchers took a cell from an adult monkey and fused it into egg cells. So this embryo allowed to develop over time and then grew cells. And then its cells were grown into the culture dish. So at this time, when these cells were in the dish, they had him like basically starving and like needing the nutrients that cells need to develop and grow. And right at the midpoint, they got these cells, made the nuclear transfer happen, and then again, another successful way of cloning. So as we go on and on through the timeline, this is when it gets to 2013, when the same scientist and his colleagues that were from the previous monkey experiment that was done in 2007 come together and are trying to find, different ways of cloning, but bringing it into humans. They put so much emphasis on cloning on humans because, like I said before, monkeys are our closest relatives. So they saw it as, why not try cloning on the monkeys first before we try it on humans? But this did bring controversy between 2004 and 2005 because people were like, people had different opinions and different perspectives of how cloning should be. But this is how cloning has been developing over time. I'm pretty sure they're still finding new ways as of today to clone, to get genetic information from, whether it be from animals or even from humans, and make different ways of making the same person be exactly the same. Thank you.
Hello, I'm Austin, and today I'm going to tell you some of the pros of cloning. One of the pros of cloning includes genetic cloning. Through genetic cloning, we are able to remove the bad traits within us, such as hair loss and our height. With cloning, we can also receive desired traits, such as green eyes or a darker skin tone. Another pro of cloning includes organ replacement. With cloning, we can recreate organs that usually require a long wait list. With this ability to clone organs, we are able to replace organs without a long wait list and ultimately save more lives. Another pro of cloning includes reproductive cloning. With reproductive cloning, we can allow same-sex couples to have children with the same genetic traits. Reproductive cloning also allows parents with either bad sperm or bad eggs to have children with the same genetic traits. Finally, cloning also benefits animals and plants. Through cloning, it allows us to save endangered animal and plant species that are endangered of going extinct by helping us reproduce them. Cloning also allows us to recreate species that have gone extinct in the past.
Hi, my name is Gabriela Paz, and I will be talking about the cons of cloning. So, to start off with the cons of cloning, there would be a lack of diversity in humankind, which could then lower the human race's ability to adapt. There would also be a lack of diversity in the world, leading to everyone looking the same, which would then lead to the less variation and less opportunity to create new varieties in the future. Also, there would be people who clone desired traits, which would lead to malpractice because people would begin to clone what they want instead of what they need. If a clone is susceptible to disease or changes in environment, then all clones would be susceptible. With everyone basically being susceptible, that would lead to having humans unable to adapt to new viruses, vaccines, and maybe even environments if they decide to move around the world. This would all decrease the value of human lives. So, with the cons of cloning, it all just comes back to having a human life less valuable because everybody would be the same person or be the same person twice. And that's not what we want in this world.
My name is Meghan Abila, and I'm going to talk about where cloning is today as of 2018. So, cloning is far more intricate and far more advanced than previous scholars thought ever possible. We see things like cloning losing its more taboo status because things are being cloned more regularly. For instance, celebrities who are cloning their deceased pets is something that is becoming more prevalent today. But I think the biggest question is cloning humans, and it's kind of the hottest topic right now. And so, according to ScienceDaily.org, there are some people who are already drawing up sort of a legally binding contract that constitutes the cloning of humans and what is allowed and what isn't allowed. And I think that with this, we must ask ourselves, what is the reason for cloning humans? So, a lot of people think that cloning humans is good because we will have essentially another set of healthy organs or blood marrow or plasma or bone marrow. And although that is true, these humans that are being cloned, what are their rights? The Constitution doesn't kind of, directly draw an artificial human's rights and what they are entitled to. And morally and ethically, is it okay to create another being just to kind of take their organs out and give it to someone else? Another kind of concept we must ponder is religion. Is it okay for humans to essentially create more life? And how do people feel about that? And so with great cloning comes great responsibility. There are essential questions that scientists must ask themselves today. And so maybe these are questions that we'll have answered when this document comes out. Maybe these questions will be answered tomorrow. I think that cloning is such a big subject and there are so many logistics and it's such a broad thing and it's so cool and so amazing and phenomenal that the future of it is very, very bright.